We start at 3 o'clock. So, all right, perfect. I think now it's live on YouTube as well. So welcome everybody. My name is Arzu Çöltekin, and many of you may have crossed my name one way or another through ISPRS activities. I co-chair ISPRS working group on visualization, augmented and virtual reality with Victoria and Sidonie, whom I think are around as well. Hi, Victoria and Sidonie. Normally we do activities around these topics, so it's geovisualization, augmented and virtual reality, but we also have a strong interest in concepts from vision because visualization theoretically grounds itself quite often on vision. So we have a lot of interest in perception, cognition, and what people see and what they don't see. So this theoretical grounding in vision to be able to design visualizations well brought me to contact with someone at the MIT at some point, and her name is Ruth Rosenholz, and some of you may actually cite her work on visual clutter because that's, I mean, she does a lot of different kinds of work, but this is something that our community picked up. And in one of the visits at the MIT, I ended up actually crossing paths with Daria, and her interdisciplinary interests are fascinating because she did an undergrad as an engineer and took an interest in vision herself. So basically studied as an aerospace engineer and oceanographer with a PhD at the MIT in collaboration with Woods Hole Oceanographic Institution. Usually looking into biological and computational vision underwater. This is one of her sort of niche interests or the depth expertises, and as well as using off-the-shelf RGB cameras for scientific imaging underwater. As some of you might know, ISPRS also has actually an underwater imaging working group. So they have also advertised Daria's talk after this was announced, and I think there would be a number of them also here. Hi, everybody from the underwater imaging working group. And Daria has professional, technical, and scientific diving certifications, conducted underwater field work in multiple parts of the globe, Bering Sea, Red Sea, Antarctica, Caribbean, Northern and Southern Pacific, and Atlantic. So you can imagine that she's been really a global citizen all over the world. And her native Aegean. Now, talking about her native Aegean Sea, she's from Izmir, I think, Daria, right? And this is, I'm also from Turkey, so this was also a connecting point. As soon as I have seen her name and last name, I'm like, okay, we probably speak the same language. And one thing that I wanted to mention, because as an oceanographer, that's a wonderful name to have, because Daria in Turkish could mean ocean. It can mean also like universe, something a bit larger, but we also say Daria when we mean ocean, so it's a perfect match. One other thing that I also wanted to mention about Daria, which is there's a lot one can say, is that she also was nominated and in fact awarded Blatavnik Awards for Young Scientists in Physics and Engineering for her postdoctoral research for resolving a fundamental problem in underwater computer vision. The reconstruction of lost colors and contrast, which led to the development of an algorithm called See-Through, which is also an app, I think. And at the moment, she's a senior lecturer at the University of Haifa, just located about a month ago. And her research lab is located at the Inter-University Institute of Marine Sciences in Eilat, Israel, so just next to the Red Sea. So I'm really looking forward to hearing what Daria has to share with us. Thank you very much, Daria, for making the time. I know that time is everything these days, so we really appreciate it. And looking forward to actually giving the floor to you, because I talked already too long. Please go ahead.
Arzu, thank you so much, first for your invitation and then for this warm welcome. I will go ahead and share my screen. And sorry, again. And if you can confirm that you see it, I'll go ahead and get started.
Yes, we can see the screen. And maybe a very quick note, sorry for interrupting you, for the audience. We are using the chat feature to get your questions. You might see both Q&A and chat. Please use the chat to collect the questions at the end, so we can have a Q&A session also with your involvement. Thank you very much, and back to you, Daria.
Okay, thank you. Good afternoon or good evening, everybody. I know everybody's all over the globe, so wherever you are, I am very happy to be invited to speak to this audience because I think some things that are so natural and common in my work will be very interesting and new to you. And then later we can exchange ideas about what's natural and common in your domain of research that are actually new or difficult or challenging to me. So I have a very colorful presentation to you today, and I'm going to talk to you about the very important role of photogrammetry in the color reconstruction of underwater images. So let's get started. Here's basically how I structured today's talk. I'm going to tell you briefly about myself. Arzu did already, but I just want to mention a few more things. And then I'm going to spend 5 minutes talking about things I'm actually not going to talk about today, which is my visual ecology projects in the ocean. And then I will move on to the core of today's talk, which is going to be centered around why is imaging colors in the ocean such a hard problem? And what role does photogrammetry actually play in helping solve this problem? So briefly, as Arzu said, I was born and raised in Turkey. And by training, I'm an aerospace engineer. I'm a graduate of the Middle Eastern Technical University. I also did my master's in aerospace at MIT. And at that time, I was specialized in thermodynamic efficiency of aircraft engines, which is not the most exciting field. And it sort of made me question whether I wanted to stay in academia after a master's or not. So I thought to myself, okay, let's go to the world and make some money and see what the business side of things is like. And I actually got a job in consulting, in IT consulting for a finance company. And I was young, single, no family, no children. I worked a lot, I traveled a lot, I made a lot of money. But some of you may remember in 2008, there was a small crisis in the finance sector. And in parallel with that, I was realizing that I was spending way too much time indoors in an office, and I was missing the changing of the seasons because I was stuck in front of a computer all the time. So I thought to myself, you know, I have a good background, I'm educated, I can choose other paths. What do I want to do with my life? What kind of job do I want to have so that I can look forward to showing up every morning? And the answer to that question was a job that puts the ocean in my life every day. I didn't want the ocean to be something I experience in my life 2 weeks a year on vacation, but I wanted to work with the ocean every day. So at that time, I made the decision to go back to school, get a PhD, and I actually earned my PhD again at MIT jointly with Woods Hole Oceanographic in mechanical engineering and oceanography. I graduated in 2014 and I never looked back. After that, I was a postdoc in Panama, in Israel, in Germany, in the US. For a short time, I worked in Florida as a research scientist at Harbor Branch. And at the moment, I actually just started a tenure track job at the University of Haifa in Israel, but my lab is on the southern part of Israel in Eilat on the Red Sea, and this is our campus. And I'd like to take these 10 seconds to advertise that I am looking for outstanding students who are interested in problems of vision and imaging underwater. It could be biological vision or it could be computer vision, because my work is very interdisciplinary. So if this is something interesting to you, you know, some folks who would be interested, please pass on my email, which I'm happy to share after the talk. Okay, so with such a weird background in ending up as a ocean scientist, what do I actually work on? Well, I work at the intersection of ocean optics, computer vision, and visual ecology. So working in ocean optics also means that I also work with ocean biogeochemistry. So most of the time I am in a multidisciplinary project acting as the bridge between these fields. And today I'm going to start, as I said, briefly by telling you about my visual ecology projects, just a couple of them. And hopefully you'll get a sense of what somebody with an aerospace engineering background does with animals and their vision in the ocean. So the two projects I picked out for you are cuttlefish camouflage and the visual systems of sea turtles. I want to start with this video that was taken by my PhD advisor, Dr. Roger Hanlon, in Grand Cayman in 2005. So you probably have seen this because it's been all over, even before social media was a thing, it was all over the internet. It keeps coming back in different viral posts. But as I play it, just watch the center of the scene. Just watch the rock as the camera approaches the rock. And in a very short time, you realize it's not actually a rock, it's an octopus. And the only reason why Roger knew that this animal was there, because he had been following it with this camera. And because he's a cephalopod, he's a scientist studying these animals, he was attacking the animal with this camera to capture the coloration. Now it's going back in reverse. Look at the point between its eyes. That's where the coloration starts, and then it spreads throughout the skin. And very quickly, you will see that it becomes almost indiscriminable from its background. This is really remarkable because they're not matching only the color, they're not matching only the pattern, they're taking their skin from 2D to 3D, they're changing their shape, and they're doing this within milliseconds. But to me, what's more interesting and impressive than doing this in milliseconds is the fact that they're colorblind. As far as we scientists have been able to check in their eyes, they only have one photoreceptor, which means they don't have color vision. And that's very interesting. They could have other mechanisms to sense color. We don't really know. Nothing has been proven, validated. There are theories out there. So a relative of octopus is cuttlefish. You probably know squid. We eat a lot of squid. Cuttlefish, octopus, squid are all cousins, and cuttlefish is the one that has a white bone inside. Sometimes this bone you can see in bird cages that the birds pick on. So another relative of octopus that I have been working with for a long time is cuttlefish, and I'm interested in understanding how, when they have the ability to change their body pattern, their camouflage, how do they decide which pattern to choose, which camouflage uniform to wear? And for this, I put them in small arenas like this, give them ridiculously unnatural backgrounds, and sometimes also natural backgrounds. But the thing about the unnatural backgrounds is that I know their statistics exactly because I designed them, right? So I put the animals into these little arenas and time lapse them until they find a stable pattern, they choose where they're gonna sit, and they camouflage. And then I try to relate what they ended up wearing to what was in the background. And this is a long project that's still ongoing. This one in the middle with the sine wave had a really hard time deciding where to go. So this is sort of on the camouflage side. Now moving on to some very interesting adaptations in the eyes of marine turtles. Now, us humans, we are trichromatic, right? We have 3 photoreceptors in our eye, or photoreceptors in our eye. It's sensitive to 3 different parts of the electromagnetic spectrum. Sometimes we call these red, green, and blue, like we know from cameras. But turtles have a 4th photoreceptor, which is sensitive to the UV. So already their chromatic world, their color world is much richer than ours. But on top of that, if you take the eye of a turtle out, cut it open, look at the retina, you will see that there are these yellow, orange, and red oil droplets scattered all over the retina on top of the photoreceptors. These are called colored oil droplets. And turtles are not the only ones that have this. Some fish and many birds, and in history, even some mammals had them, but then they lost it. But turtles have retained them for millions of years. So here's a cross-section of the cone in a turtle eye, those finger-shaped organelles, and then the yellow oil droplet sits right there. So an important question is, what advantage do these oil droplets give to turtles? Because, as I said, the other animal group that has them is birds. And birds live on land under broadband daylight. And birds themselves are very colorful. They use these colors in communication, in mating, in signaling, in camouflage. So there's a lot of reasons why these little organelles would benefit birds. And in fact, theoretical work showed that what oil droplets do for birds is enhance their color discrimination. So because of these oil droplets, their visual system can discriminate more colors, which makes a lot of sense. Again, birds live on land, there's a lot of light, there are a lot of colors, there's a lot of roles of those functions of those colors, but turtles are not colorful. They live in ocean habitats, which become less and less colorful with depth. And other than the coral reefs of the world, ocean habitats are not necessarily that colorful. So what advantage could oil droplets be giving these turtles? This is the question I wanted to understand. And one way to tackle this question is to see the underwater world like a turtle. So how are we going to do that? Because we can't use off-the-shelf cameras, which are designed for our eyes, our trichromatic eyes, and also we don't have these oil droplets. So the solution was to build a brand new camera that sees the world like a turtle. And as a part of my previous postdoc, I built this in the lab of Tali Trebic, also at the University of Haifa. This camera has a sensor that has sensitivity to 16 parts of the electromagnetic spectrum, not just 3 like our everyday cameras, but 16. So it's a multispectral sensor. We packaged the sensor with a simple Linux computer, put some buttons to interface with the diver, and put it in an underwater housing that we could take to 100 meters. And it became the turtle eye camera. Here you see its first dive, its first test dive in the Red Sea. So now we have an instrument with which to see how turtles might be able to see. That's cool. But what's cooler is what the footage looks like out of this camera. So here on the left, you see the data this turtle camera captured, the hyperspectral data cube that I processed exactly according to the visual system of a real turtle which has photoreceptors and oil droplets. And on the right, you see the same footage processed for the same photoreceptors, but without the oil droplets. And already, even though I said turtles aren't colorful, oceanic habitats aren't colorful, already you see the difference between the chromatic richness, we can say, the number of colors, the diversity of colors between the two visual systems. So this is quite cool. This work is still ongoing. It's not finished yet. And it's only an engineering model of the turtle's visual system. So do turtles actually see like this? And what does this mean to them? Those are ecological questions. We haven't answered those yet. So that's just a quick summary of my visual ecology projects. I'd be happy to take questions about them at the end. Now I want to move on to the core of the talk. Why is imaging colors in the ocean such a hard problem? And how does photogrammetry make it a bit easier? So perhaps we should take a second and talk about the role of colors in the ocean. First of all, in terms of the ocean water, colors can give us tremendously important scientific insights. Many of you work in remote sensing. You definitely know better than me about the field of ocean color, where we try to sense the color of the water remotely from instruments on satellites, and then try to make out which organisms which type and concentration of organisms result in that color, so that we know, is there phytoplankton, how much phytoplankton is there, how often is that changing, how is it changing throughout the globe. So there's a lot of information that we can get from the color of the ocean water. But just the color itself actually is not enough. For us to get scientific insights, there was another very important milestone. And that milestone was the ability to sense the color of the ocean water for the entire globe from space, remotely. And again, I'm sure you guys are very familiar with this map, but this is the first time we were able to use pseudocolors, or we were able to map the ocean color at a global scale. And it took about 10 years to build this map. And when scientists suggested this, that this can be done, that from space we could see the color of the ocean and then we could make sense of it in the 60s, people ridiculed them. They said that will never happen. We'll never be able to see the ocean from space and at this scale. And if we did, we're never going to be able to make sense of what's in the water. But of course, today, you know that we do this every day at very high resolutions. And this has really revolutionized oceanography. The ability to sense the global ocean from space has absolutely revolutionized our understanding of the ocean. Because if you think about it, before our ability to do this, scientists would go to sea, take a measurement here on a boat, travel 2 kilometers south, take another measurement, and between those two measurements, assume that everything varies linearly and smoothly. But you look at the ocean from space and you see that nothing is linear and nothing is smooth, and the scales of change, these eddies, these currents, these boundary currents, everything can be from very small to very large, both spatially and temporally. So to summarize, the color of the ocean water is an extremely important property of the ocean that gives us great scientific insights. But then there's people like me who don't care at all about the water, but want to see what is on the ocean floor and want to see the true colors of what is on the ocean floor. And to be able to see that, the fact that the water has color is a big problem because it masks the color of everything else on the ocean floor. So what can we do about this? Every photo we take looks like this, blue or green or yellow and washed out with low contrast, and color contrast is nearly zero. So before I move forward, I want to take a second and I want to define what I mean by color, just so we are all on the same page. So color, very generally, is a sensation that's defined as the interaction between the reflectance of a surface, which is a physical property of a surface that determines how much of the incoming light spectrum it reflects. It's the interaction between the reflectance of that surface, which is a physical property, and the available ambient light as sensed by a specific observer, a specific sensor. So I'm going to make this definition even easier. And for the rest of my talk, when I say color, you can just assume that I'm always using the same sensor. So we're going to kick sensor out of this equation. So very simply, when I say I want to see the true colors of everything in the ocean, what I really mean is I want to see the reflectance of everything in the ocean without the bias of the color of the water, which essentially is the color of the ambient light. So that's what I mean when I say color. Now, let's quickly review how an image is formed underwater. This diver wants to take a photo of this octopus, and unbeknownst to him, the photo he will take will actually be a sum of 3 different photos. The first layer, out of his camera, he will only get 1 image, but that image will be actually a superposition of 3 different images. The first image will be formed with the light that comes from the sun. It travels to the skin of the octopus, and then it reflects from the skin directly to the camera without scattering along the shortest path or along what we call line of sight. And this is really the most interesting and useful image for the diver. It's a photo of the octopus, but the light maybe will attenuate along this path a bit. But then this photo will be degraded because water molecules and particles suspended in the water will actually scatter light in ways that we don't want, ways that they degrade images. For example, there are these very exaggerated plankton organisms in the water, and light from the sun will reach the octopus, will reflect from the skin of the octopus and hit one of these really exaggerated floating particles, and then scatter back towards the camera. So now what happened is, instead of a crisp image of the octopus, a second image of the octopus will form, but it will be very blurry. It will be like a glowing version of the octopus. So this is a degrading event. It's not that important. The major component of image formation that actually degrades underwater images is this one that I now show with the red arrows. Light will come from the sun. It will scatter from these exaggerated floating particles. It will never travel to the skin of the octopus, rather directly scatter to the sensor. So what does that mean? It means that it's going to create a colored layer of fog, no information about the octopus whatsoever, just a colored layer of fog that occludes the diver's image. So that we call backscatter. The useful signal, the one I showed with the blue arrow, is called the direct signal. And the glowy, ghostly image of the octopus, which is not such big of a problem, we call forward scatter. For the rest of this talk, we're going to ignore forward scatter, and I will now give you a couple of examples of the direct signal and the backscattered signal. And soon it will become clear what the link is to photogrammetry and why these are such big problems. So here's our diver again. He's on the bottom right of the image, and now he wants to take a photo of the color chart. And the color chart is right above the starfish. We use color charts a lot in our work because these are surfaces that are spectrally calibrated. In other words, we have measured their reflectance. And when we take them underwater and then take images, we can assess how the ambient light and the water conditions change the surface properties, and then we can work out some parameters that we need. So where the diver is, on the bottom right of the image, he's way too far. When he takes an image of the color chart, his image will be pretty much blue because that annoying layer of backscatter, that fog, will be so dominant, there will be zero signal of the color chart. When he gets a bit closer, his image will actually start to contain the color chart. You will be able to see the color chart, but still the backscatter or the fog will be dominant enough that it will not be a sharp image of the color chart. Only when the diver is right up to the color chart, in other words, he's very close to the scene, then his photo will almost entirely contain the color chart. The layer of fog will be almost zero, and the direct signal will dominate. So to put it into perspective, how the fog grows and how the colors attenuate is a strong function of distance. So there's one takeaway from this slide, and that is, if you are interested in underwater photography, your best bet is to always get as close to your subject as possible, minimize the backscatter, and then you can do your editing, your post-processing, and bring out the colors. But if you're too far and there's too much of fog between you and the scene, forget that photo. Now, this was a simulation just to get you acquainted with the terms direct signal and the backscatter. Now I want to show you a real example. Here's a real color chart that I mounted in the middle of the ocean. And then I swam to this color chart from 8 meters back all the way up to 30 centimeters. And I photographed it as I went. Now you see to the left, from the left to the right, as the distance gets closer, there is a very annoying occluding layer of fog. It's almost like the color chart is at the end behind a curtain that really lowers the contrast. To show you how bad that fog is, now I'm computationally going to remove the fog. And you see now all the charts at all distances are actually, they have the same sharpness. So this comparison shows you the effect of backscatter in the real world. I'll take it one step further. Now we remove the backscatter, but we still have attenuation of colors. And I will computationally put those back. If you focus only on the patches of the color charts, you'll see that each patch has been, the color of each patch has been restored consistently across distances. So essentially, this is how images are formed underwater. This is exactly how images are formed in air, except the particles in air have sizes that are very small compared to the wavelength of light. So this occluding layer of fog does not become significant until we have actual fog, where there are large particles in air and there's fog and we can't see anything. To summarize, the problem of color reconstruction that I work on and many others work on is simply the following. Given an image that's all blue, low contrast, and you know nothing about where it was taken, how it was taken, which camera it was taken with, which depth it was taken with, from what distance, nothing, you don't know anything. Can you restore the true colors in that scene? And maybe some of you will look at this, especially those of you interested in photography, and you will say, well, can't we just white balance that? No. And the reason is the following. Because white balancing does not work underwater. Let me show you why. There are 5 color charts in this image, and actually a 6th one that's being held by a diver all the way back. And all of these color charts have a white patch. And now when I extract the white from all of them and put them side by side, you will see that the white is different at every distance. So how are you going to white balance the scene? You can only choose one white value, and that will be maybe okay for the chart in number 2 position, but it won't work for the chart in number 6 position. So because of distance dependent attenuation of light underwater, and because that layer of fog, the backscatter, grows with distance, we cannot use white balancing. So we have to use algorithms that know distance. And here is where the difference between image processing and computer vision is. Image processing is an area where you work on the pixels of an image to make them better, to filter them in some way, but you know nothing about the contents of that image. In computer vision, you have information about the scene structure, or maybe about the objects in the scene. You have 3D scene structure, or you try to get 3D scene structure. So you might be wondering, you know, we have computer vision in everything in our lives today, in our cell phones, in supermarkets, in public transportation, in even some ovens, we have computer vision. So how come we don't have robust algorithms that fix the colors of underwater images for us already? I mean, this is 2022. Why? And to give you the answer to that question, I have to take you back 200 years. Okay, 200 years ago, roughly, was the time that the first photograph was made. And not much long after, actually only 30 years after, the first underwater photograph was made. And this wasn't by taking a camera in the water, but rather putting a camera in a wooden enclosure and lowering the enclosure. And again, around that time, in just early 1900s, first measurements of sunlight spectrum at hundreds of meters of depth in the water was measured. And this is very impressive that this was done back then. And then came World War I. And of course, wars are a time where a lot of technology is being developed very fast. And when the war is over, that technology suddenly finds new uses in the public domain, and it becomes a subject for research, which is exactly sort of what happened between World Wars I and II in terms of visibility in the atmosphere and in the water. About 1924, we had worked out the physics of visibility in the atmosphere, which is more or less pretty close to what we use today, maybe with some improvements, but the basics were there. And this work after World War II ended, accelerated in the underwater domain as well, and most notably in the visibility lab of Siebert Duntley, which started at MIT and then moved to Scripps. Around that time was the first underwater camera, the Calypso, maybe some of you will know. And then Duntley published his seminal Light in the Sea paper, which we still refer to and cite heavily today. In 1970s, the first digital camera was invented, and this is what it looked like. So those of you, if you can see, there is a cassette tape on the side of it, because that's how we recorded the images. And in the 80s, with the advance of the computers and faster hardware, we started to be able to simulate underwater scenes in a computer. So that was first done also in the visibility lab by Ben McGlamery. Then we improved this. Jules Jaffe, who's a very important figure today at Scripps in underwater imaging, he refined McGlamery's model. But here's the key point in all of this nonlinear timeline. In 1999, Sri Nayar and Srinivas Narasimhan published this very important paper, computer vision paper, called Computer Vision in Bad Weather. And bad weather here refers to fog, rain, snow, weather events in the atmosphere. And what they did in this paper is they took the radiative transfer equation, which is a massive physical equation that governs what happens to light in a scattering medium, and they simplified it for the terms of the atmosphere. And then they further simplified it for the three-dimensional space of a consumer camera, the RGB space of a camera. And then they came up with an image formation model that described how vision, as far as a camera goes, works in air. And here's the turning point. For the next 20 years, researchers used this equation, which was originally derived for conditions in the atmosphere, for underwater images until 2018. So what happened in 2018? Finally, we had an equation for image formation that was derived for the ocean. And this was me and my advisor, Tali Trebic, at the University of Haifa. After a long period of not being able to fix the colors in underwater images, we finally went back to fundamentals and showed that the equation that was commonly being used was actually designed for the simplistic conditions of the atmosphere. Image formation in the ocean is a much harder problem. An atmosphere is only a subset of it. So once we have this equation, it became very clear why we didn't have algorithms that fix the colors in underwater images. Because just think about this for a second. On the left, there is a scene in air with fog. It's a pumpkin field. And on the right, there is an underwater scene. So there's very obvious differences here. In the fog, we have visibility that we lose in hundreds of meters. And the color of the fog is generally white. In contrast, in the ocean, there is never a day that the color is white, right? You've never seen this before. It's light always attenuates as a function of wavelength. So there's this blue curtain. And also visibility is lost in such short distances. And all of this is because attenuation in the atmosphere is more or less generally wavelength independent. Here you see on the x-axis wavelength, and on the y-axis attenuation coefficient for both the ocean and the atmosphere. And it doesn't matter where this data comes from, it's just to demonstrate that in the ocean, there is never a time when attenuation happens without dependency on wavelength. So this brings me, I know it's not popular to show equations, but I'm going to walk you through this equation and we don't have to remember it. So this brings me to the difference between what was wrong with the atmospheric model and what's different about the oceanic model. All you have to remember from this slide is that an image, which is I in both cases on the left-hand side, has an attenuation coefficient that governs how the colors are lost. On the bottom, where we have the model for the ocean, we show that we need to have two different coefficients. But in the atmosphere, it was always assumed that one coefficient could govern everything that's happening in an image. And more importantly than the coefficients, this is not the focus of this talk, notice this Z. Z is distance. Remember when I showed the examples from the beginning that everything was changing and getting worse as a function of increasing distance? So that's Z. And whether you're in the atmosphere or you're in the ocean, the distance is a very important parameter to recover lost colors. So now that we derived this equation, we have to do something with it. An equation on itself is not valuable. So we went ahead and designed an algorithm that uses this equation. And the algorithm was designed for RGBD images. D here means distance or scene depth, not the depth from the water surface, but the depth between the camera and the scene. So we called it see-through. And this algorithm uses the new equation, takes the Z or that distance as input to put the colors back. I'll just walk you through a few examples of what putting the colors back means. Here's a typical underwater image taken in the Florida Keys. And when we use the distance to each pixel and calculate the parameters we need, now we can put the colors back. Essentially, this is what this scene would look like in air. Here's another scene. This time it's a reef from Papua New Guinea. And when we use the distance to each pixel and remove the water, you see that the reef is actually ridiculously colorful and full of different animals. So these are corals and corals are animals. Another example from the Red Sea. We can remove the water and put the colors back. Again, this is working because we know the distance to each pixel. We get stunning results. Nothing stops us from doing this, let's say in a swimming pool. And now we can see not only the colors of the bathing suits of the swimmers, but also their skin tones. Okay, so this is essentially color reconstruction. Where does photogrammetry come into play? Distance. Remember how important distance is? So how do we get distance in an underwater scene, in an underwater photo? First of all, in oceanic and global scales, we get distance using acoustics because sound waves travel much, much, much further than we can get imaging with optical sensors. But with optical sensors, there are specialized sensors like LiDAR, like many of you in remote sensing use, but actually in the domain of the ocean, it's not that common. It is very expensive. The ones that are commercial are on the order of $300,000. Here you see in the photo one that we have recently used. This is about $100,000 and it's quite large. So we can't really use LiDAR. We can use ordinary cameras to get distance. Either we need to have a grid of multiple cameras and synchronize them geometrically and then extract the scene structure, or we can use a single camera and take multiple images with it, which is what brings me to photogrammetry, because this is the cheapest way for any researcher or marine lab out there to use the cameras that they already have, go out to the sea, collect multiple images that are overlapping a certain amount, and then create a 3D model of the scene that they are interested in. And from the 3D model, here's our equation again. This is our raw image. you export a depth map from the 3D model that you made using structure from motion. And this is the Z that this equation is looking for. So the critical piece in color correction, the hardest thing to get, because it's not one per image, it's one per pixel. Every pixel has a different distance, is the depth map. And until we could do this with photogrammetry, there was really no easy, cheap way to do it for pretty much any average marine lab, because it's already expensive to work at sea and you can't afford the LiDAR. So there, unless you use stereo cameras, there was really no way to get the distance. This is the color reconstruction story, but we use photogrammetry in marine science for many, many, many different applications. The majority of them being for coral reef science, but the fish community, here's a nice histogram of a survey done by Orion McCarthy from Scripps and San Diego, basically showing the tool is becoming more common. It's mostly used for mapping of the seafloor and monitoring changes before and after events like a big storm, but also it's increasingly being used as a conservation and policy tool, because you can show how things are changing and you can say, well, maybe we need to protect this area. Maybe we need to close this area to fishing because such and such. So it's not easily, the images you take underwater and then to build a 3D model of it is not so easy, precisely because the water degrades the images too much, but it's increasingly being used. So I want to show a couple of examples of applications of photogrammetry that are not color reconstruction. For example, these slides are from my friend Matan, and he's comparing the structural complexity of coral reefs in 3 different locations in the world. And for this, he's using photogrammetry and 3D models. Similarly, he's looking at the diversity of corals in a coral reef, and he's segmenting them, he's annotating them, he's extracting their volume, their sizes, and then actually monitoring that across time. As I said, photogrammetry is an extremely useful tool to monitor the state of the seafloor, especially before and after weather events. Here on the left, there is actually a reef from Eilat, where I am, before we had a big storm 2 years ago. And on the right, you see the same reef after the big storm, and a lot of the corals are dead. Of course, the natural next step for any photogrammetry underwater is to do the 3D models with corrected colors. It's a bit of a chicken and egg problem. So first you collect the images, put them into your photogrammetry software to get the depth maps. You take the depth maps, go back to the images, and then you make color corrected images, and then you make a color corrected mosaic. So where does all of that leave us? I tried to give you a very brief glimpse of the very hard problem of image formation and color reconstruction, and how we use photogrammetry for different applications in the ocean. But my personal interest is answering this one question: why are there colors in the ocean? Because now that we have a way to look at the seafloor, and the 3 images you see here are the seafloor from different places, why are these colors there when the color of the water is going to mask it anyway for any viewer? So what's the function of these? I don't know the answer. I'm very excited to dedicate my life to finding out. So if this sounds interesting to you, please consider getting in touch and maybe working with me in my lab. That's all I had. I think I'm okay on time. So thank you very much, and I'm happy to take questions.
Thank you very much, Daria. That was amazing. As you can also see from the messages on the chat, people are very keen on your, and we are really happy to learn something from you. For the sake of Q&A, I would like to invite those of you who would like to Yeah, I see already one hand raised. I have a couple of questions, but I'm going to save mine and start with the audience. And if I if there's time at the end, I might reach you later. Cheryl, can you help me with that? So if somebody's raising hand, can they also speak?
Sorry, we are in a webinar mode, so we can only use the chat and the Q&A box for the questions.
All right, so those of you who are interested in asking a question, it sounds like you will have to type it. So when you are typing the questions, I'm sorry about that. I think it's just about broadcasting restrictions that we have at the moment with the setup. So please write them, and I see that a lot of very nice feedback coming in. The topic is interesting, the talk is inspiring, and excellent lecture, beautiful presentation, amazing talk. So there's a lot of very, very nice positive feedback, and I'm very pleased to see that. If when you're typing your question, maybe I start with one question, Daria, if you don't mind.
Sure.
Maybe there's actually a similar question, perhaps. I was just going to ask this color board you used for calibration, and I wanted to ask, how do you choose the what primary colors or what kind of color? Because there's colors are so many and different. It can be a huge selection. Which ones do you decide that must be in the calibration process? And the audience question from Morgan Liu is also related to the color board. Maybe you can handle them together. And this one says, do you use the color board to calibrate the wavelength dependent backscatter coefficient? So that's a more specific question. So
I'll go ahead and answer both of them. First, how do you choose the color chart? The answer is very simple. I use basically one of the 2 or 3 in the market that is waterproof. And that means that I didn't specifically choose the colors, but whatever color chart I buy, I measure the reflectance of each patch. So I know exactly what I'm working with. So as long as the chart is waterproof and you can print your own chart, no problem, just measure the reflectances so that you have that information. So basically it doesn't matter, you can use any color chart. As for Morgan's question, do you use the color chart to calibrate? The answer is no. In the algorithm, so let's put it this way. The color chart you saw in the photos is there because after we do the color correction, we need a way to assess how well did we do. And we need ground truth, but we cannot take the habitat out of the water for the most part. So the way we do that is we place color charts in one or multiple places and only check our success, our performance on those places, which is actually very simplistic, but that's how it's done. The algorithm itself does not need a color chart because it would be very impractical. For example, if you're imaging with a robot, you would have to drop a color chart, go pick it up. If you're imaging a fish, run, push the color chart behind the fish. So that wouldn't work. The algorithm works without a color chart.
Okay.
Hopefully that answers.
I believe it does, and in the interest of time, I think I'm going to have to take some other questions. So Morgan, if there's like deeper...
By the way, Arzu, I'm happy to stay a bit longer if folks, I know it's Friday afternoon or evening, but I have time, so we can take all of them.
That's excellent. Cheryl, we announced 1 hour, right?
Yes, but I think we can extend a bit if you would like.
Okay, perfect. So I think if Daria has time, we can add another 10-15 minutes if there's more Q&A. And I see that there's a number of questions. So those of you who have to be elsewhere or have other engagements, obviously you're welcome to leave at the end of the hour or anytime you are ready to leave. But if there's some flexibility, that would be excellent to be able to address the other questions as well. So let me see. I think the next one in terms of the timeline might be the one in the Q&A. There was one in the Q&A. Sorry, I had to switch to the cell phone. If you, I think you can see what I'm doing here. Because the battery ran out of the laptop and I didn't have enough time to run for the charger. All right, so I see that I think maybe Todd's question is the next one, and there are two of them from Todd Rubin. And Todd says, could undersea animals use their depth perception to see colors underwater? Basically an enhanced vision system, and continues, oh, actually it's actually a corrected version of the same question. So basically, yeah, how do underwater animals I mean, you talked about turtle vision. I guess that's what inspires the question. So how do the underwater animals handle the color vision? Do we have some information, or do you know, did you think about that, Taya?
I wish I could answer that deterministically. The truth is we know so little. So we have all the physiological information, right? We can cut open the eye, we can look inside, we can see the cells, we know the eye size, we know the physics. But in terms of 3D, of course, the ones that have two eyes are using 3D. There are some very simplistic animals that have many eyes. For example, scallops, which are bivalves, they have 200 eyes, 2 retinas, a lens that doesn't do anything, and then a micro mirror array that focuses the light. So there's all kinds of crazy visual systems, and very few of them we know how actually they utilize the visual information in the water. And that's actually a great area to go into if anybody's interested. But just to answer, yes, I mean, animals have stereo vision, they see depth, but how do they do color correction? Do they do color correction? Is it more of an adaptation? I can't answer.
All right.
Maybe ask me again in 10 years.
Perfect. And maybe after today's talk, some excellent postdoc comes and helps you answering the question.
Yes.
Perfect. So there's another one in the Q&A, and then I'm going to go back to the chat. I think that was also timeline-wise quite correctly aligned. Hayat Kadi asks, are the color constraints the same with videography as for photography?
Excellent question. Thank you so much for this question. I didn't get into the technical details, but because we are using a physical equation, we must work with raw images. And what does raw image mean? It means what the sensor captures has a linear relationship to the light in the field. Now, this is the case for almost every consumer camera that shoots RAW. The image is linearly related to the light, so our equations apply. When you come to video, because there is the need to record so many frames per second, first of all, the camera wants to compress those frames to be able to write at sufficient speeds. But second of all, because every single camera we're using was actually designed for imaging on land, almost every camera applies its own in-camera, proprietary, undocumented enhancements, nonlinear corrections to the image before it compresses it, and those changes get baked in to the camera frame, and now the linearity is lost. So the short answer is, if you have one of those very high-end video cameras that shoot raw video, yes, we can apply the same rules, no problem. But once the camera enters its irrelevant processing for atmosphere into the images and compresses them, there is no going back. So if you're in marine science, if you need to use underwater data, please shoot raw to the biggest extent that you can.
Okay, thank you. I think we can maybe take the next one in the Q&A that I skipped. I think I skipped one, and then I'll continue on with the next the other questions. Nicholas de Kock asks, does the algorithm work if one were to use underwater strobes as part of their camera setup?
Excellent question, Nicholas. Thank you so much for this question. I love this audience. Yes. So again, we go back to the equation that I showed, which I passed in like 10 seconds. Another time, maybe Arzu, we can do a 2-hour session on just the equation. This equation, Nicholas, is designed for natural light because natural light is very well characterized. If you were using strobes, we would need to know the spectral distribution of your light as well as the spatial distribution of that light and the geometry relative to the camera. If we could calibrate that, with slight modifications, we could use the same equation and then we could develop an algorithm. But you can imagine how variable the strobes and the geometry is from person to person. And even in one dive, like a shark or a barracuda can come and kick your strobe and the geometry will not be the same anymore. So then we would need new calibration. So this is the problem, not problem, but difficulty with physics-based models. You have assumptions and you have to work within those assumptions. So the short answer is what I showed you today does not work with strobes.
All right. Thank you, Daria. I think I'm going to read the next one now. That's a sort of a specific one. Peter Shi is asking, in my experience with underwater photography, SFM packages failed. What is your success rate?
I can't tell you how many days and nights and hours of my life I wasted trying to do mosaics underwater. If you were in clear enough water and if you could get close enough to your scene, you could build a nice model. But if you were too far or if the water was too murky, they will not align. You will not be able to build a model. You have to do all kinds of tricks. This is not the way to go. SFM is not the future of 3D modeling for underwater imaging. I think it will be LiDAR. So that's my experience. I hate sitting in front of the photogrammetry software, but as you see, it's the best solution and we use it now, but hopefully not for long.
All right. That's interesting what you said about LiDAR, but I don't want to steal time from others. So I will say that, for example, Yoon asked if you could explain the equation again, but I feel like that might be, like you said, 2-hour lecture. Yoon Zhang, I think that we're going to see about the equation another time. I was trying to put the publications from Daria on our GeoViz VRAR Twitter account. Maybe this helps you a little bit. For the moment, I feel like we should go for the other questions and then come back for the equation another time.
And if you guys email me or I can send you, there's a few videos on YouTube that explain how the method and the equation works. Those may be more accessible than the papers. So I'll share those with you as well.
That would be perfect. We can make, we also have a little blog. We could make a blog post and share with all the audience from today with the links, embedded links to the publications and the videos that you might share with us, like a little proceeding from the talk, if you don't mind. That might help.
Absolutely.
All right. So then you have a few more compliments. Somebody says, very impressed with the presentation. Also working on the domain from TU Dresden. Melanie offers more compliments. There's so many of them. Yeah, you should save this chat and put it in the back of your CV.
Thank you.
And that's very nice. And then I think the next one, so this is Frank Niemeyer. Frank asks, how do you realize the scale in your underwater 3D reconstruction?
Very good question. In most of our examples I showed, you saw the color chart. So even though we don't use the color chart for color correction, it's always there for later. And I simply just use the scale of the dimensions of the color chart. That being said, everything I showed you today could be done without knowing the scale, just relative scale.
And I will, I'm just kind of messenger here, so I'm reading the next one. Evangelos Alevizos, I'm sorry if I'm destroying your names, Fox, is asking, apart from distance Z correction, how about the backscattering from particles or dissolved matter?
Yes, so this backscattering is a huge problem. As I said, Evangelos, that's a very good question. So we calculated because we know from the physical model what its relationship to distance is, and it's an exponential relationship, and there's two parameters that govern that exponential shape. So the algorithm has a way to work that out. That is also in one of my videos, or I can send you the papers. We don't actually calculate the physical backscattering coefficient because you cannot. It's what the physical got projected into the camera sensor space. So we calculated in the camera sensor space and then back it out. So we calculated, in short. I'm not sure if that was helpful, but I can gladly talk to you about this more later.
Yeah, Evangelos, if that wasn't really exactly the answer you were looking for, please follow up with
Please.
us. I'm happy to facilitate if you can't reach Daria directly. The next one is Alessio Calantropio, and he is saying, thank you for the amazing presentation. How did you manage in obtaining a depth map for the image of the swimmers in the swimming pool, since I believe single camera photogrammetry wouldn't work in that case?
Okay, hi Alessio. So this is a very good question. There's a trick you can do in cases like this where the subjects are moving, just shoot in burst. And when you shoot in burst, you click the shutter once, but you get like 3 to 5 images all at once. And in very clear water, that's enough. So that's how I did that.
So you do have multiple images.
Still, yes, exactly.
All right. And Emanuel Rocco is asking, when you showed the multispectral picture, what was the hypercolor cube, or how did you transform the multispectral picture into RGB?
Right. So Emmanuel, you are right. You have the hyperspectral data cube, and then you have the spectral response of the camera or the turtle or the visual system you want to work with. And according to the image formation equation that's relevant to that case, air or water or whatever, you essentially multiply the two together. So there is a full-blown equation that says you can put the sensor and the visual information together and you get the colors into RGB.
Perfect. Do you have some more patience? I have a couple of more.
Yes, please.
All right. So Mehmet Büyükdemircioğlu is asking, amazing work again. And my question is, he says, is the generated 3D point cloud model is geolocated in a real world coordinate system? If so, what is the accuracy? I suppose like when you capture these underground information, do you always know the location? Do you have a sort of GPS-like thing that functions underwater? That's how I understand the question.
Yes, another good question. Thank you, Mehmet. So I don't specifically work with georeferenced images because my world is, single images in the water. However, I know that there are systems that you can calibrate, and you take a GPS point in certain parts of above the water where you're going to sample underwater, and there's some dead reckoning in the water. When you come out, you take another GPS point, and then you can have rather accurate georeferencing. I haven't done it, so I don't know the accuracy, but I know that people who monitor the same site over and over again have a system, even a commercial system, that they can do that with.
Thanks, Daria. There's Norm next. Norm Wog Vincent says, you showed that the wavelength dependent attenuation coefficients depend on the setting, turbidity, presumably depth. So how do you find these without calibration?
That's an excellent question. That is the 2-hour talk. We need to do another time. No, I'm sorry. Have a look at the papers or the videos. And if not, shoot me an email and I'd be happy to talk in detail. But so basically, that's the whole method. That's another talk.
So it's fairly long answer otherwise. All right. So thanks and answers from Hong Kong, from Morgan again. And then we have Iftan Song. Thank you. Iftan says, depth estimation and color restoration is a chicken and egg dilemma. Would it make more sense to combine revised image restoration model with underwater photometric stereo to estimate geometry and restored color together?
So yes, you found this is a great point. The answer is yes, but so stereo, of course, you put 2 cameras together, you can put more cameras together. After calibration, it can give you depth in real time, but actually it's generally quite inaccurate for distances further away from the camera, like the distortions are too high. So if you are talking about rather close distances, this is definitely a solution and it can work in real time with the color correction, yes. But I still think the way to go is LiDAR.
That's interesting. So LiDAR works as well in underwater as it does in the air, would you say?
Yes, with slightly different principles in engineering, but yes. And big order of magnitude difference price tag, but yes.
All right. All right. So we have a few more. Thank you. Thank you for the answer. Thank you for Daria and organizers. You are all very welcome. Thank you so much for being such an active audience. And we have Marinos asking actually the same, I would say, similar question about how do you estimate backscattering as well as attenuation coefficients? Is it possible to elaborate? I sense the answer is come back with the longer answer, right?
It's a 2-hour talk, Marinos, but briefly remember that we have a physical equation which tells us how everything changes with distance and the water coefficients. And basically we do some optimization per pixel to find what satisfies that equation. But again, that's a 2-hour talk.
Excellent. So Peter says he will try to take the photos closer. I will
job.
try
Excellent.
closer to the object. And then we have one of our student consortium members. I don't see exactly which one of you is saying that. But a suggestion, perhaps an experiment suggestion with the iPhone that comes with LiDAR, which has a 5-meter range. Would that vary underwater? It is cheap and affordable. Could one experiment with that? Do you think it's reasonable to think about?
I wish, but the wavelengths they use, so that's infrared, it will go about 5 centimeters in the water before going completely extinct. So unfortunately not.
All right, so then I think I have some more feedback from the people who asked for further elaboration of the equations from Yuzang and Evangelos. They would like to be in touch and receive more of the information that we promised them. And I think I'm getting more of the thank yous and excellent webinars. And
Great.
I think that's about it. Did I miss any questions in the Q&A, or let me see if there's more here. Maybe I think we are good. I think the people caught up on the Q&A versus chat. So this may have brought us to actually more or less a wrap-up point. The talk is recorded, and I think that it may be, if you are fine with that, Daria, I don't remember if you already asked your explicit permission, we might issue it also on YouTube.
Absolutely, sure.
That's perfectly good. And then I will, from our working group, I would be happy to follow up with a blog post that features this video and supplementary materials. Maybe we disturb you with 1 or 2 more questions as to other materials that you mentioned now. And I think with this, I'm again very thankful for you for that you made time for us. And thank you very much for an active participation from all of you. It was